Welcome to our comprehensive guide on the development and validation of dissolution procedures. Dissolution testing is a critical quality control tool in the pharmaceutical industry, ensuring that drug products release their active ingredients appropriately. In this video, we'll walk you through the essential steps, apparatus, and validation strategies for robust dissolution testing. Whether you're new to the field or looking to refresh your knowledge, this series will provide a structured overview, starting from the basics and moving into advanced considerations. Let's begin by understanding why dissolution testing matters and how it fits into the broader context of pharmaceutical development. Dissolution testing serves multiple purposes in pharmaceutical development. It assesses the batch-to-batch quality of drug products and evaluates the physical stability of formulations. By simulating the drug release process in the body, dissolution testing helps ensure consistent therapeutic outcomes. The procedure must be reproducible, discriminating, and rugged enough to handle variability in formulations. Additionally, it should be transferable between labs and have low variability in results. These qualities make dissolution testing a cornerstone of quality assurance in drug manufacturing. Understanding sources of variability is crucial for reliable dissolution testing. Variability can arise from the formulation itself, such as differences in excipients, hardness, or friability. Artifacts including air bubbles or equipment inconsistencies can also impact results. The physico-chemical properties of the drug substance, like solubility and pH sensitivity, play a significant role. By identifying and controlling these variables, we can design more robust dissolution procedures that yield consistent and meaningful data. Using the right dissolution medium is foundational for accurate testing. The medium should reflect the physico-chemical properties of both the drug substance and the dosage form. Factors like solubility, pH, buffer capacity, and the presence of surfactants must be considered. The medium should be inexpensive, readily available, and ecologically acceptable. Common options include purified water, simulated gastric or intestinal fluids, and buffered solutions. The goal is to mimic physiological conditions as closely as possible for meaningful results. Composition of the dissolution medium can vary widely depending on the drug and its formulation. For example, aqueous organic solvent mixtures, hydroalcoholic solvents, and various buffer systems are used to achieve the desired pH and solubility. Surfactants like polysorbate or sodium lauryl sulfate may be added for low solubility compounds. The volume of the medium, typically between 500 and 1000 ml, also affects dissolution rates. Ensuring sink conditions, where the drug remains soluble throughout the test, is essential for accurate measurement. pH of the dissolution medium is critical, especially for drugs with pH-dependent solubility. For modified release MR formulations, pH ranges from 1.2 to 7.5, while immediate release IR products use 1.2 to 6.8. Surfactants, such as bile salts or SLS, can enhance the solubility of poorly soluble drugs. The concentration of surfactant must be carefully controlled to avoid artifacts. By adjusting pH and surfactant levels, we can better simulate the drug's behavior in the gastrointestinal tract. Volume of the dissolution medium is typically set between 500 and 1000 ml, with 900 ml being common. For certain apparatus, larger volumes up to 4 liters may be used. Maintaining sink conditions is vital. This means the drug should remain fully dissolved throughout the test. If the medium becomes saturated, dissolution slows and results become unreliable. Proper volume selection ensures that the test reflects the drug's real-world performance. Air bubbles can interfere with dissolution testing, especially with gelatin capsules. De-aeration methods include heating the medium to 37-41 degrees C, sonication, vacuum treatment, or filtration. These steps remove dissolved gases that might otherwise form bubbles, affecting the dissolution rate. For gelatin-coated products, enzymes may be added to address cross-linking issues. Proper de-aeration ensures that the test environment is consistent and free from artifacts. Vitro in vivo correlation, IVIVC, links laboratory dissolution results with actual drug absorption in the body. By using biorelevant media that simulate fed and fasted states, we can better predict how a drug will perform. The rate-limiting step, whether dissolution or permeation, determines the choice of medium and test conditions. IVIVC is especially important for formulation development and understanding food effects on drug release. Several types of apparatus are used in dissolution testing, each suited to different dosage forms. Apparatus 1 basket and apparatus 2 paddle are common for solid oral dosage forms. Apparatus 3 reciprocating cylinder and apparatus 4 flow-through cell are used for modified release products and poorly soluble drugs. Specialized equipment like mini paddles, bead-type systems, or rotating bottles address unique formulation challenges. The choice of apparatus depends on the product's characteristics and regulatory requirements. Sinkers are used to prevent floating dosage forms from rising in the dissolution vessel. They come in various sizes and materials, such as stainless steel or platinum wire. The choice of sinker depends on capsule size and shell type. Proper use of sinkers ensures that the dosage unit remains fully submerged, allowing for accurate measurement of dissolution rates. Accessories like mesh screens and corks may also be used for specific formulations. Agitation rate, measured in revolutions per minute (RPM), is a key parameter in dissolution testing. Too low an RPM can lead to inconsistent hydrodynamics, while too high can cause turbulence. Typical rates range from 25 to 150 RPM, depending on the apparatus and product type. For suspensions, specific mesh screens and RPM settings are used. Proper agitation ensures uniform mixing and reliable results. Well-designed dissolution study includes multiple time points to capture the release profile. For immediate release products, sampling at 5, 10, or 15 minutes is common, with a single-point specification like, not less than 85% in 15 minutes. Extended release products require early, intermediate, and final time points to ensure complete release and detect potential dose dumping. At least 3 time points are recommended for robust analysis. Visual observation is a valuable tool during dissolution testing. Proper lighting and clear visibility in the bath allow for the detection of phenomena like uneven particle distribution, air bubbles, or adhesion to the paddle or basket. Documenting these observations with photographs, videos, or sketches helps identify issues that may affect the test outcome. Visual checks complement quantitative data and support troubleshooting. Sampling can be performed manually or automatically. Manual sampling involves using syringes and cannulas, while automated systems offer routine performance checks, maintenance, and cleaning. It's important to ensure that sampling probes do not alter the dissolution rate. Automated sampling provides consistency and reduces human error, but both methods require validation to confirm they do not introduce bias. Filters are used to remove undissolved particles from samples before analysis. The pore size typically ranges from 0.45 to 70 micrometers. It's important to compare filtered solutions with centrifuged or unfiltered samples to ensure no drug is lost or adsorbed. Proper filter validation ensures that the measured concentration accurately reflects the dissolved drug, not just what passes through the filter. Centrifugation is sometimes used to separate undissolved particles, but it's generally not preferred due to potential interference from excipients or drugs. Instead, filtration is favored for its simplicity and reliability. Analytical methods like HPLC or spectrophotometry are then used to quantify the dissolved drug. The choice of separation method impacts the accuracy and sensitivity of the assay. Assays for dissolution testing can be automated or manual, using techniques like HPLC or spectrophotometry. Automated systems improve speed and reduce solvent use, while manual methods offer flexibility. The assay must be validated to ensure specificity, accuracy, and precision. Placebo interference, linearity, and recovery are key parameters to assess during validation. Placebo interference occurs when excipients or other ingredients affect the assay result. To measure this, placebo solutions are prepared and analyzed alongside standards. The interference should not exceed 2% to ensure specificity. Calculations involve comparing absorbance values and concentrations, ensuring that the assay accurately measures the drug without significant background noise. Linearity ensures that the assay response is directly proportional to drug concentration across the expected range. This is tested by preparing solutions from below the lowest to above the highest expected concentrations. The correlation coefficient, R2, should be at least 0.98, and the y-intercept should not differ significantly from 0. Organic solvents used to enhance solubility must not exceed 5% unless validated. Accuracy is assessed by spiking known amounts of drug into the dissolution medium and measuring recovery. The acceptable range is typically 95.105% of the amount added. For delayed release forms, special validation is needed for the acid stage, with a limit of not more than 10%. These checks ensure that the assay can accurately quantify the drug under all test conditions. Precision is evaluated by repeated measurements of the same sample, calculating the relative standard deviation, RSD. Repeatability checks ensure that the method yields consistent results within a short time frame. Intermediate precision involves testing across different days, analysts, or equipment. Acceptance criteria are set for allowable differences, typically not more than 10% at lower dissolution and 5% at higher dissolution levels. Robustness assesses the method's resilience to small changes in test conditions. Parameters like medium composition, agitation rate, temperature, and pH are varied to ensure the method remains reliable. For HPLC, factors such as mobile phase composition, buffer concentration, and flow rate are also tested. Robustness testing confirms that the dissolution procedure is not overly sensitive to minor variations. Stability testing ensures that both standard and sample solutions remain within acceptable limits during analysis. The response should stay between 98% and 102% of the initial value. Factors like container material, temperature, and light exposure are considered, especially for unstable solutions. Regular checks prevent degradation and ensure accurate results. Spectrophotometry is a common analytical technique in dissolution testing. Automated spectrophotometers require routine performance checks, maintenance, and cleaning. Cell alignment and the absence of air bubbles are critical for accurate readings. Path lengths typically range from 0.02 to 1 cm, and acceptable standard error and linearity must be maintained. These steps ensure reliable quantification of the dissolved drug. Automated sampling systems offer consistency and efficiency, reducing the risk of human error. However, manual sampling remains valuable for flexibility and troubleshooting. Both methods require validation to ensure they do not introduce significant changes in dissolution rates. Proper cleaning and maintenance are essential for both approaches to maintain data integrity. Acceptance criteria define the limits for dissolution results, ensuring product quality and regulatory compliance. Specifications are set for each time point, such as not less than 85% dissolved in 15 minutes for IR products. For extended release forms, criteria cover early, intermediate, and final time points. Meeting these criteria confirms that the product performs as intended. Standard operating procedures, SOPs, and thorough documentation are essential for reliable dissolution testing. SOPs cover routine checks, maintenance, cleaning, and performance validation. Proper records ensure traceability and support regulatory audits. Consistent documentation helps identify trends, troubleshoot issues, and maintain high standards in the laboratory. Common issues in dissolution testing include air bubbles, uneven particle distribution, and adhesion to apparatus surfaces. Visual observation and proper documentation help identify these problems. Adjusting parameters like agitation rate, medium composition, or sinker use can resolve many issues. Regular maintenance and validation keep the system running smoothly and ensure reliable results. In summary, the development and validation of dissolution procedures involve careful selection of medium, apparatus, and analytical methods. Robust validation ensures accuracy, precision, and reliability. Regular maintenance, documentation, and troubleshooting are key to consistent results. By following best practices, you can ensure that your dissolution testing supports product quality and regulatory compliance. Thank you for joining us on this journey through dissolution testing essentials.